The nicotinic acetylcholine receptor, NACHR, is a ligand-gating ion channel involved in the neurotransmission. It catalyzes the opening of the ion channel in response to the binding of the neurotransmitter acetylcholine and nicotine, allowing ions to flow across the cell membrane. Nicotine, like ACH, is a nicotinic receptor agonist. The binding of nicotine and ACH to nicotinic receptor causes a conformational change that either opens or closes the receptor's ion channels, thereby changing the receptor's functional state. Activation of NACHR leads to depolarization of the postsynaptic membrane, propagating the nerve impulse. In the inactive conformation, 6PV8, the receptor is color-coded with yellow, representing chain A, and orange, representing chain B. This helps visualize different structural elements within the receptor. In the inactive conformation, the active site residues, including residues from the transmembrane helices and loops, are oriented to bind ligands, but are not in an optimal position for catalysis. AT1001, the blue molecule, inhibits the receptor catalytic site. It is a high affinity and selective alpha-3 beta-4 nicotinic acetylcholine receptor antagonist that blocks nicotine administration.
The active conformation 6PV7 is superimposed over the inactive conformation 6PV8 to compare their structures. To highlight structural differences, corresponding regions in both structures are color-coded similarly. In this pentameric ion channel, A3B4 are the domains with the residues involved in the interaction. Slight changes can be observed overall across the two models as the conformations change from inactive to active forms that allow for the flow of ions. The active site residues undergo conformational changes in the active conformation, allowing for optimal binding and catalysis. These residues may shift positions or orientations compared to the inactive conformation. When nicotine binds, it's enveloped in a nest of highly conserved aromatic residues, namely Y93 from loop A, W149 from loop B, Y190, Y197 from loop C, and W59 from loop D.
In the active conformation, the active site residues interact with the nicotine substrate, color in pink, through hydrogen bonding, electrostatic interactions, and hydrophobic interactions, facilitating ligand binding and channel opening. Hydrogen bonding occurs between polar residues in the active site and functional groups of nicotine, especially the nitrogen atoms found in the pyridine ring and pyrrolidine ring, facilitating precise positioning of the ligand within the binding pocket. Electrostatic interactions between the charged residues and nicotine contribute to the complex stability. Additionally, hydrophobic interactions between nonpolar residues and hydrophobic region of nicotine further enhance ligand binding affinity. These interactions collectively ensure proper orientation and recognition of nicotine by the receptor, leading to channel opening and subsequent ion flux across the cell membrane.